Tubers are storage sites for energy and nutrients in plants. Plants use these tubers to survive in the winter and dry months when there's not much water. The tubers in a potato plant in particular are what grows in the ground and what we eat. This video shows the process of a tuber growing. The tuber is what is underneath the ground in the soil. Potatoes originated in the Andes Mountains. The climate in the Andes is known to change drastically and in short distances. Being in this climate caused the potatoes to evolve short-day dependent tuber formation. In order to understand tuberization in potatoes, scientists bred 2,000 individual siblings from one specific parental cross. They used genetic markers to identify recombination locations in these potatoes.
Recombination produces a new combination of alleles in a two-step process. First, homologous chromosomes are paired in meiosis. Then, new molecules of DNA are created by the breaking and rejoining of DNA strands. The STCDF1 gene is a homolog on a chromosome of the potato. There are three different varieties of this gene. The first, STCDF1.1, is a homozygous and late maturing allele. STCDF1.2 and 1.3 are both heterozygous and early maturing alleles. To demonstrate proof of early maturing, the experimenters took both an early maturing and late maturing plant and overexpressed the allele STCDF1.2. In this scenario, both plants tuberized very quickly, even in the long day conditions. The control plants not subjected to STCDF1.2 allele could not tuberize during the long day conditions. The experimenters concluded that the loss of the C-terminal end in the 1.2 and 1.3 alleles impairs the interaction between proteins and complexes that repress tuber formation, and therefore the tubers form quickly. Unlike alleles 1.2 and 1.3, allele 1.1 has the C terminus. Thus, the repressive complexes can interact and deter tumor formation. By truncation of the C-terminal end of STCDF1, specific proteins affect the protein-protein interaction and repression activation of proteins, but not the DNA binding ability. This provides strong evidence for the link between STCDF1 and STCO1/2.
When monitoring the protein interactions over 24 hours in both the late maturing plant and the early maturing plant, the late maturing plant, they detected a peak in protein levels between 10 a.m. and 4 p.m. in the afternoon because the C-terminus of the proteins in the late maturing plant is intact, so the proteins can act normally and inhibit the formation of tubers. On the other hand, the early maturing plant had much lower levels of protein interaction throughout the day because the proteins are not responding as much to the changes in the day length or time and therefore cannot inhibit the formation of tubers. STCDF1 is linked to three other genes. First, it works with STCO1-2, which, with the phytochromes, either suppresses tuberization or activates it based on the day length and the amount of sunlight the plant is getting. It does this by regulating STSP6A and STSP5G, which signal for tuberization. STCO1 either activates STSP5G during the long days to suppress tuber formation, or it downregulates STSP5G during the short days. By downregulating STSP5G, then STSP6A is activated and this causes tubers to form. In graph E, the activation of the STSP6A gene, which when upregulated causes tubers to form, peaks a lot more for the early maturing STCDF1.2 and 3 plants. Conversely, in graph G, the expression of the STSP5G gene, which suppresses tuber formation, peaks for the late maturing STCDF1.1 plant. Now we know that based on the phenotype of the plant, whether early or late maturing, it acts differently with the STCO and the STSP5G and 6A genes. Then, the STCO gene measures the amount of sunlight to determine the season and communicates this by either activating the STSP5G gene to inhibit tuber formation or activate the STSP6A gene to form tubers.
Under short day conditions, days when potato plants receive little sunlight, no STSP5G is detected in late or early maturing potato plants. This is because potato plants tuberize because they have to, due to short day conditions. During short days, STSP6A is expressed in both STCDF1.1, STCDF1.2, and STCDF1.3 plants, but is much more prevalent in STCDF1.2 and STCDF1.3. Under long days, STSP6A is highly expressed in 1.1 as compared to 1.2 and 1.3, with the early maturing genotype. To tie this information together, these results indicate that STCDF1 acts as an indirect inducer of STSP6A by downregulating the STSP6A repressor, STSP5G, through STCO1 and STCO2. In conclusion, each of these genes work together to determine the expression of tubers in potato plants.